Good morning, good afternoon, or good night, whichever the case may be as you watch this. Welcome again. I'm going to try to be fast, talk quick. I'll save announcements for emails. There were no assignments for this weekend other than to post your Flipgrid. With that, I want to continue. Before watching this lecture, you might want to go back to Chapter 2 and review what hydrolysis and dehydration synthesis reactions are and how water is formed and necessary for each of those. We'll refer back to it here in this, but that's a kind of a minimum before coming into here. So what I want to talk about is the structure of proteins, and then we'll also talk about the structure of DNA in the next lecture, and then we'll have a third lecture that is protein synthesis, where both DNA and proteins come into play. So what you're looking at here are real representations of actual proteins. This is the way protein chemists and journals often and typically refer to them. Space filling models like these. 3. This is kind of a ribbon model. You've kind of outlined the sequence of amino acids, but these are actual structures. A Nobel Prize was given for this one right here in the upper right corner. Does anybody know what it is? Anyone? Anyone? Hemoglobin. Yep. So you got a polypeptide, 4 polypeptides that come together to form one functional protein, and that's hemoglobin. This one down here, anyone? Anyone? That's keratin, or collagen, excuse me, it's collagen. So you got 3 identical polypeptides, just like you have 4 up here. These are only 3, forms kind of a rope. That's a structural protein. You have enzymes here. So let's go on with this. Proteins are very important. We talk a lot about proteins because they do essentially everything in the cell. They're involved in almost everything. And the only reason we can say almost everything is maybe there's something that occurs, minor reactions without proteins. But for the most part, anytime we look and talk about reaction, it has to do with proteins. So they serve as catalyst. That's enzymes. They speed up chemical reactions, primarily in most metabolic reactions of the body. Also, they are support proteins, again, collagen, keratin, in your skin and hair primarily. So when grandma or your Aunt Leona or whoever it is pinches your cheeks or gives you an Indian arm burn, your skin kind of pops right back into place. And over time, they get weaker, and the elastin in your skin gets weaker, and so it begins to sag. But otherwise, your skin is pretty structurally sound because of these 2 proteins. Carriers and transport, you'll see for circulating transporters in the blood, you have the hemoglobin. You also have transporters carry like iron in the blood and such, receptors and binding for defense. So a lot of defense mechanisms here. You're talking about immunoglobulins, antibodies. Those are all proteins circulating in your blood made by cells. Histocompatibility context. Each of these will be discussed to some degree later in cell biology. But obviously actin and myosin, which we'll learn about in this class this semester for contraction, signaling hormones. You could say regulation, like insulin, regulating blood sugar. It's just one example of hormone signaling. It's also a protein. All these functions are carried out by various types of proteins. So proteins should become an idea in your head that they are a very diverse structural group, which allows them to perform very functionally diverse, very diverse functions is what I mean to say. So structure dictates function. We have diverse structure. We're going to have diverse function. You mess with the structure, you're going to mess with the function. That's where a lot of disease and mutation comes from. I'm going to try to slow down here. I'm trying to beat the clock. So we're going to go on to the next slide. Here's the structure again. To remind you have a centrally bound lower right-hand corner, carbon, bound to a hydrogen. And right here you have the amino acid group, or the amine functional group, as listed in your textbook, NH2, which can be NH3 in its ionized form. And over here you have the carboxylic acid, or the carboxyl group, COOH. All right. This can also be ionized. And typically, it's the hydrogen from this hydroxyl group, OH, that can come bind over here to this nitrogen, become NH3. Now, that's not all being reflected in this picture here. The point of this picture is just to show you the formation of water. So this is an example of dehydration. We dehydrate the 2 molecules, and you notice it's H2O. So you dehydrate, you pull out water, and you synthesize a new bond. Dehydration, synthesis. This bond is called a peptide bond. And so now where there was 2 individual amino acids, you have one dipeptide, 2 amino acids formed from the carbon-nitrogen bond. And just kind of the study of this picture will show you that. So as a monomeric group, and again at this point from your readings, you might want to review the difference between a monomer and a polymer, but monomers are building blocks of polymers. Mer means piece. Mono means one. Poly means many. So you have one piece, that would be an amino acid, that's one piece. Many pieces would be polypeptide, and that's after you string a whole bunch of these together. We referred to some of these in class, and I think you saw the ball stick models. Yes, you did. So a protein can be one or more polypeptide chains folded and bonded together, and they're large, complex 3D shapes. So, structural overview. Named amino for this group, acid for this group. Definition of an acid: any compound that when added to water donates hydrogen, and that's what happens in an aqueous solution. Aqueous meaning water-based solution. And as that hydrogen becomes free, nitrogen will take it up because it has an affinity or electronegativity for these hydrogen, or positivity I should say, and you have the NH3, and bonds can be formed as a result of that. But what is it that makes each amino acid different? The previous slide said there are 20 different amino acids that are essential. So what makes a difference is this radical group right here. There could be one of 20 different radical groups. This forms the backbone of the chain, but the overall structure and function of the folding of that protein is a function of this radical group. So we're just gonna, you don't have to memorize these structures, you don't need to freak out, but I do want you to answer this question to yourself. So you'll notice these are non-polar amino acids. What does that mean? Well, if they're non-polar, they don't have charges, so they do not interact well with water, therefore they are hydrophobic. So here are some examples: glycine, alanine, valine, that are hydrophobic. You'll notice they're all CH3. These are methyl groups, methyl functional groups, also discussed in your textbook. So they're very stable. These ring structures tend to be hydrophobic, aromatic amino acids. So CH3, so there's not going to really be any interaction with water. They will interact well with each other, but they will not interact much with water, and that's okay for now. And here's a group of polar amino acids, and looking at the same characteristics, you'll see that OH, so that can donate that hydrogen. OH, it can donate that hydrogen. SH can donate, that's a sulfhydryl group. All these have an extra hydrogen that can be lost. This side of threonine is CH3, but if you look at this side, it can also become charged and ionized. Down here, we have acidic and basic groups, and that is because they're native negative charges and native positive charges. You can see the NH3 and NH2, just as we did the backbone over here of the amino acid group. Again, you don't have to memorize all these names and which ones are which. Just understand that there are polar and nonpolar amino acids, and that's going to affect everything about what their possible function is, where they're located, why it is that their structures can perform so many different things. All right, there is one thing I'd like to point out. There are only 2 amino acids. You'll see cysteine right here as an SH, and then there's another, if I can find it. Let's see, I have a slide that's talking, here we are. So there are some sulfur-containing amino acids. Cysteines form cross-links with each other. So this will become important when we talk about amino acids interacting with each other, because many times amino acids interact with each other through hydrophobic interactions or ionic bonds or hydrogen bonds. Recall that hydrogen bonds, ionic bonds, and hydrophobic interactions are all weak bonds. Covalent bonds are very strong because they share electrons. Well, these disulfide bridges are covalent bonds. So 2 sulfurs or a sulfur and carbon can form a strong covalent bond, and that will definitely put a permanent structure in whatever protein it is. So you see a lot of this in hair. A good example is hair, keratin. there are some versions that have the sulfur. And so if you happen to have wavy hair or really curly hair, you probably have a lot of these sulfur-containing amino acids. They tend to kink up like that. It used to be a fad to have a permanent, right? So if you had really straight hair and you wanted to put some body into it or wave, and you'd go get this stinky smell and stuff sprayed all over your hair, and you go through a whole process at the beauty salon. And what they're doing is adding sulfur to it and then rinsing it out, and you're causing it artificially to kink up. Those who have the wavy hair, you just need to break those covalent bonds. And the way you guys do that are with those hair straighteners. And you put the hair straightener on, right? And you comb it over and over and over, trying to add heat energy to those bonds to break them so that your hair will go straight. And then, of course, once you put water back in it, the polarity attracts them and you're back to your wavy hair. Anyway, so building proteins. Peptide bonds, here we are, dehydration synthesis, linking the NH group of one amino acid to the carboxyl group of another. That is, linking the NH2 to the COOH of another. And they're end-to-end. If you flip-flop this amino acid, no bond would occur. So it can only occur in left-handed amino acid molecules. And again, the same picture here showing dehydration synthesis. And here's a peptide bond, arranged a little bit different. So molecules in 3D space and time don't come with the top and bottom labeled. They're left and right. So for our reference, we have to call it N-terminus and C-terminus. The N-terminus is that side of the protein that has the N, and the C-terminus is with the C. You'll see this again shortly. So building proteins N to N. Here it is, N-terminal, NH2, C-terminal, COOH. This is called carboxyl. This is called the amine group. So the repeated sequence of NCC is the polypeptide backbone, and it always grows in only one direction. It is not random, and the enzymes that put this together know this. So even though these are dehydration synthesis reactions, they don't just randomly occur because, oh, look, we have an extra hydrogen, we have an OH. Nope, an enzyme is necessary, a protein enzyme, to link these amino acids together. So here's our N-terminus, and here's our C-terminus. And what you need to know then with all these amino acids and the structure and the alignment is that protein structure determines function. Function depends on structure. The twisted, folded, coiled, random-looking shapes are unique for the particular function they have, and there's some of the same proteins we discussed. Here's an example of an enzyme, and all this structure, all to get this active site right in here. It's pretty amazing. So, if protein structure is so important to its function, and the function depends on that structure, then it all starts with the order of amino acids. My question for you to think about, what determines the order of the amino acids? If those amino acids have to be in such an order that the hydrophilic and hydrophobic amino acids can interact together, then it's very important that they're lined up correctly. What determines that order? If you guess DNA, you're right. So what we call the primary structure, and there will be 4 structures of this. So the primary structure of a protein is the order of amino acids, that is the sequence of amino acids in the chain. So here's a long polypeptide sequence, and they're numbered at every 5, 10, 15, 20, you can see. Here's the N-terminus, down here is the C-terminus, so we call that kind of the front, if you would. So the amino acid sequence is determined by DNA. And so when Francis Francis Crick and James Watson discovered the structure of DNA, they noted in that original seminal paper that it has not escaped our attention that the sequence of DNA does fit and may determine indeed the sequence of the protein. And that was a big question for years. Well, for 100 years we knew proteins were involved in heredity, or so we thought, and there's a big argument for about 1900 to 1940s. And it was a very slow process of experimentation to acknowledge that nucleic acid was a hereditary material. Everything up to that point was proteins, and still Nobel Prizes are given for proteins. Only one Nobel Prize was given for DNA, interestingly enough. But that's because proteins do everything, and the structure is very important. It's hard to get those structures through crystallography. But bottom line is, nonetheless, that sequence of amino acids, which is ultimately responsible for its structure and function, was determined by DNA. So any slight change in the amino acid sequence can affect the protein structure and its function. Likewise then, one mutation or change in DNA may alter that sequence of amino acids in the protein. So you can see now maybe where the DNA mutations lead to malfunctioning proteins. So even just one amino acid change can make all the difference. Best example of that is sickle cell anemia. So here's a sequence of amino acids, 1 through 7, at least it's in the first 6. So glucine here was replaced by valine, and that was just 1 point mutation in the DNA caused 1 amino acid difference. And now instead of red plump donut-shaped cells of red blood cells, you have these sickle shapes. That's sickle cell anemia. I don't have a picture for it, but it's very similar, and the same with cystic fibrosis. One mutation causes a malfunctioning protein in the cell membrane, and you cannot secrete salt. So water will not follow, so you build up really thick mucus. That's a discussion for another time if you're curious. So going on to the secondary structure of a protein, it's the local folding. So folding along short sections of the polypeptides that are near each other. So these are interactions between 2 adjacent amino acids. So you can see here's one amino acid, here's another amino acid, and then right next to each other there's just enough. So these dotted lines represent hydrogen bonds. The solid lines represent covalent bonds. So here's the hydrogen bonds, and it's just a weak attraction, but it's important enough. And they're close enough that they form these alpha helices and beta pleated sheets. And you see this motif in every protein practically. So this is what we call the secondary structure, local folding, as told by the alpha helix and the beta pleated sheets. Those structures are possible because of the hydrogen bonds between the R groups. Excuse me. Here's another picture of the secondary structure. And again, we just, to get rid of all the molecules, make it a little prettier, you can begin to see how we just kind of draw a line down the backbone and it forms these coils or helices and these beta pleated sheets. It's an amazing thing. Here's an electrostatic interaction between the N terminus and a C terminus, and because it does, this kind of kinks up and forms a very specific kind of a bend. Here's a hydrophobic interaction between 2 hydrophobic R groups, 2 amino acids. It causes a slight curvature. Likewise, there's a middle ion coordination. So all these are local folding. That's secondary structure due to hydrogen bonds or weak interactions. Tertiary structure is the whole molecule folding, and that's determined by these R groups. So you notice this bend might have come because of ionic bond, hydrogen bond, and this because of hydrophobic interaction. Those are methyl groups. So hydrophobic interactions cluster in groups away from water, and van der Waals interactions, short, temporary, but they're enough to cause these bends and twists and helices. So look in the middle here, and this entire conformation is supported now by this disulfide bridge, which is a covalent bond right there. So that's really strong. So it's going to be hard to get rid of this shape and structure. So what you have here is, while these might have seen some local folding, you're going to have whole molecule folding here, and we'll look at another protein that helps this go by. So last, quaternary structure joins together more than one polypeptide chain. So typically, again, we've learned everything. We have tertiary, whole molecule folding, secondary, local folding due to the amino acid sequence, and primary structure, which is the order of the amino acids in the chain. So that's all the bonding. So nothing really new in quaternary structure except to say that sometimes a single polypeptide chain is not a functional protein until it's bound to other polypeptide chains. And so here's collagen, like in skin and tendons, very strong tensile strength. Here's hemoglobin again. So how are these 4 polypeptides held together? Same forces that were holding the others. This hydrophobic, typically hydrophobic interaction. And so that way they stay together because water kind of forces them together. Water has this shaping because of its polarity. And these are hydrophobic interactions. The hydrophobicity of these molecules turns in on each other, and it really actually confirms the shape of the molecule. So you should go through this slide. When you open the slideshow, it goes one at a time, and it's designed for you to kind of pop quiz yourself on the primary structure, secondary structure, tertiary structure, and lastly the quaternary structure, and reviews what determines that structure, such as amino acid sequence by DNA, hydrogen bonds for secondary structure, more hydrogen bonds, hydrophobic interactions, disulfide bridges for tertiary structure, and then all those playing again to the quaternary structure. And that's about it for this. The remaining slides are details about chaperone proteins. And this is one of the proteins that helps to guide whole molecule folding. Protein models are visualized. Just talks a little about the science, X-ray crystallography, computer modeling. A lot of Nobel Prizes, like I say, over the last 100 years, even today, being given for structures and discovering the structures of these proteins. That's all medicine. It's medicine, because if we can kind of fake a protein that's in excess or that needs to be shut off in the body, you can make a drug that fits that protein or silences it by binding to it in a special way. Can't bind to a protein unless we know the shape. And so protein shape becomes very important. Denaturing protein, well, heat can disrupt the 3D structure, as well as salt, pH, because pH is basically the power of hydrogen. So if you have a lot of hydrogen ions in the environment, all those positive charges are going to attract to the molecule and cause it to slowly unravel. Some of these can go back into conformation, but many of them cannot. Think of a scrambled egg, albumin. When that protein denatures, it is permanently denatured. So, go review protein structure, and hopefully protein synthesis will make more sense to you. All right, have a good day.